This is a film about a type of molecular Velcro, a class of proteins called lectins, molecules capable of binding to or agglutinating cells in our body, often in a blood group specific manner. Lectins are found in some very common foods, including many beans and seeds, fruits, vegetables, grains, and seafood. These molecules were given the name lectin by their discoverer, William Boyd. Boyd chose this name because it is Latin for the phrase, "I choose." Lectins are quite common in nature. 1/3 of over 3,000 plants tested showed lectin activity, and 36 of 88 types of prepared foods tested agglutinated cells of one ABO blood group or another. A lectin typically binds to carbohydrate sugars. Since the ABO antigens are themselves sugars, as many as 50% of all lectins are ABO blood group specific. Lectins are a widely diverse class of molecules, both large and small size, with any number of binding sites, and share nothing in common other than the ability to bind with carbohydrates. Some of the most toxic compounds known to man are lectins, including ricin, the lectin found in the castor bean plant. In the early 1970s, ricin was used by the KGB to assassinate the BBC commentator Georgy Markov. This was accomplished by the use of a tiny platinum pellet containing a microscopic amount of ricin, which was injected into Markov's leg by the tip of an umbrella while he was waiting for a London bus. Markov died 2 days later from massive destruction of his blood cells. Most lectins gain entrance into the body in less flamboyant ways. Some are assimilated through the mouth, others through the stomach, but most lectins interact with the delicate lining of the small intestine, a tissue richly laced with carbohydrate. This lining is characterized by finger-like projections called villi, which greatly increase the surface area available for nutrient absorption. In addition, each villi has its own microvilli, which gives the intestinal lining the appearance of a plush bath towel. The microvilli can actively absorb lectins right through their lining. The absorption of lectins by the cells of the small intestine is called endocytosis, because the lectin is absorbed in an intact form and transported into the cell as a vesicle or bubble. There it can migrate into the blood or lymph. Microvilli are quite delicate, and evidence is growing that many lectins are capable of causing damage to them. Lectins can initiate allergic reactions at the microvilli, which perhaps explains some aspects of food allergy. Many microorganisms, including the yeast Candida albicans and most species of bacteria, contain lectin-like molecules on their surface, which they use to attach to the gut wall. The combination of food and microbial lectins probably account for many types of digestive inflammation. Dietary lectins have been shown to migrate from the small intestine to the liver, the colon, and the pancreas, in addition to gaining access to the systemic circulation. This may help explain why certain lectin-containing foods appear to aggravate inflammatory conditions. In rheumatoid arthritis, the antibodies directed against joint tissue are structurally different from a typical normal antibody, in that the sugar galactose is replaced by the amino sugar glucosamine. It has been speculated that glucosamine-specific lectins, such as those found in wheat germ, attach to the galactose-deficient antibodies and amplify their effect. Perhaps this explains why many people with rheumatoid arthritis feel better when on wheat-free diets. The lectin in pokeweed is a potent stimulant to specialized cells of the immune system called lymphocytes. When stimulated by pokeweed lectin, these cells begin to rapidly reproduce. Children who have eaten the brightly colored pokeweed berries are occasionally seen in hospital emergency room with greatly elevated white blood counts, a symptom which could be mistaken for leukemia. One of the more fascinating characteristics of lectins is their ability to mimic the effect of certain hormones in the body. Because of this, they can have profound effects on metabolism. When not burned for energy, carbohydrates are stored as fat. This is controlled by the hormone insulin. Under insulin stimulation, the fat cell becomes more permeable to carbohydrate. Without insulin, carbohydrates cannot enter the fat cell and thus remain in circulation. Many grain lectins have been shown to bind to the insulin receptor, where they cause the fat cell to accumulate stored carbohydrate as fat. However, unlike insulin, lectins do not have any feedback control, so they do not turn off, resulting in a fat cell which constantly turns carbohydrate into fat. Thus, the effect of lectins on the insulin receptor may help explain why certain individuals on high carbohydrate diets have difficulty losing weight. This may be true more now than ever, since our modern grains have a much higher protein content than in years past. Increasing the protein content of grains may be good for the world's malnourished, but we are increasing the lectin content as well. One thing is certain, however, that we have only begun to appreciate the health significance of this diverse and fascinating family of molecules.